Hello, everyone, and welcome to this presentation about visual phasing, discovering the DNA inheritance of three Lehmann brothers. Now, visual phasing is an advanced genetic genealogy technique that has been pioneered over the past decade, and it can be pretty arduous um, and take a lot of time, but it can be immensely rewarding in terms of learning more about in, inheritance of DNA. And so I wanted to make a presentation going through at first some of the science, going over the biology of how chromosomes are recombined, and then talking about chromosome maps and seeing from DNA testing how this translates into chromosome maps. And then finally, the core of the presentation will be about visual phasing and how visual phasing can be a really helpful way to build out chromosome maps. So without further ado, let's start the presentation. All right, so as I mentioned, I wanted to start with some basic biology and genetics 101. And on this slide, we'll talk about chromosomes. So chromosomes are structures found in the nucleus of cells that carry long pieces of DNA. Each cell in the human body contains 23 pairs of chromosomes. So 46 total chromosomes, but 23 pairs. 22 of these pairs we call autosomes, and they are numbered by size. So we can see over here those 1 through 22 numbered by size, 1 being the largest, 22 being the smallest. These are the autosomes. And then there is also one additional sex chromosome pair. In females, they will have two X chromosomes, and in males, there will be an X and a Y chromosome. And for the females, the X chromosome does recombine like the other autosomes, but on the Y chromosome, it largely does not recombine. All right, so for each chromosome pair, as we see, you can see each of these pairs of chromosomes. One of them is inherited from the father, and we call that a paternal chromosome. And the other, is inherited from the mother, and we call that the maternal chromosome. So across all the autosomes, and also for the sex chromosomes, you will get one of the part of the pair, one chromosome from the father, one chromosome from the mother. So I wanted to spend a moment to talk about the sources of genetic variation in the human population. It's important for the genetic health of the human population for there to be genetic diversity. And there are two ways in which genetic variation occurs in the production of offspring. So what's one way that we can see genetic variation? Well, that is mutations. And what is a mutation? It's a novel heritable change in nucleotide state that introduces new variants. We see this, oftentimes when we talk about Y-DNA. Y-DNA will look at mutations, these point mutations that occur in the Y chromosome, where a letter is changed, where instead of a G you have an A, or vice versa. And we use those mutations where they occur through the passing of the next generation to better understand the history of a paternal line. And even though we talk about mutations a lot in the context of Y-DNA, mutations, especially point mutations, but even like insertions and deletions of DNA, they can occur on any of the chromosomes. But we're not going to talk about mutations today. We're going to talk about a second way that genetic variation occurs, and that is through recombination. So recombination is a process in which pieces of DNA are broken and then recombined to produce new combinations of alleles. So let's take a closer look to see how recombination occurs. So in order to talk about recombinant recombination, we have to talk about the process of crossing over. So crossing over is the process that leads to recombinant chromosomes. When does it take place? Well, crossing over takes place during meiosis, which is an important stage in the development of egg and sperm cells. So these are happening in the development of egg cells and the development of sperm cells before fertilization. So in the development, let's talk about exactly what this process looks like. And I've got a nice little graphic here that will also illustrate it as we go along. So this again is the first stage where a cell has a paternal chromosome and a maternal chromosome, and they're labeled blue and purple colors. So in the development of a sex cell, the paternal and maternal chromosome DNA strands are first replicated. So this first step we see here is the DNA is getting replicated, and each replicated piece of DNA is connected to each other through a structure called a centromere. That's right in the middle here, and they are identical copies of the same chromosome. So you see the two blues and the two purples, and we call those identical copies by DNA replication sister chromatids. So these are the blue sister chromatids and the purple sister chromatids. Now, the next stage is where crossing over takes place. So crossing over occurs when two homologous non-sister chromatids, so these are the blue and the purple, they will pair up with each other and exchange different segments of genetic material to form two recombinant chromosome sister chromatids. So what you're seeing here is that a piece of the purple DNA is getting combined with the blue, and the blue going to the purple, and they're exchanging segments of genetic material during this crossing over process. And then from there, the chromosomes and cells are divided twice. Now in this picture here, in this graphic, we're only seeing the first division, but this takes place during meiosis 1, and there will be a second one during meiosis 2. And it results in 4 haploid sex cells. A haploid cell is a cell that contains just one copy of every chromosome, not 2. These are diploids, where you have 2 of every chromosome, but we will get to a haploid state at the end of meiosis 2. And so there are basically 4 different daughter cells that take place at the end of meiosis 2, and only one of them, one haploid egg cell, and one from the father, from this one haploid sperm cell, they will combine together during fertilization. But we see here at the end of meiosis 1, after the exchange of DNA, that you'll see that there's some of the DNA has now been mixed up between the maternal and paternal chromosomes. They are no longer separated in the same way. Okay, now let's just see a broader visualization of crossing over and the resulting 4 gametes, the 4 haploid cells that occur at the end state prior to fertilization. So again, we have the replication, and then here we have the crossing over. Here's the crossing over being completed. Meiosis 1, they get divided up, and then with meiosis 2, they get further divided up, and here you have 4 different gamete cells. One of these will then be combined with the other parent during fertilization with another gamete from the other parent to form the child. Okay, so let's move on and let's talk about the takeaways from crossing over and recombination. Why is this important that recombination occurs in the formation of egg and sperm cells, sex cells? So again, to start to review, each person inherits a chromosome pair for all 23 chromosomes. 22 of them we call the autosomes, but one comes from the father, that's called the paternal chromosome, and one is from the mother, we call that the maternal chromosome. So there are paternal chromosomes across all the 22 autosomes, and there are maternal chromosomes for all 22 chromosomes. Now, the crossing over process during the formation of an egg and during the formation of egg and sperm sex cells prior to fertilization means that every child will inherit a patchwork of DNA from each grandparent. So the paternal chromosomes, for example, they contain DNA from the paternal grandfather and the paternal grandmother. And the maternal chromosomes for the child will contain DNA from the maternal grandfather and maternal grandmother. So recombinant chromosomes are the result of the crossing over process. That's the final result that we get. And they are an important source of genetic diversity in the human population as you're mixing DNA from each grandparent in the child. So this is a really nice example on the left of just how this is illustrated. So this is the dad with purple and red denoting the dad's maternal and paternal chromosomes. And then we have the mom with the yellow and the blue containing the maternal chromosomes and the paternal chromosomes for the mother. So these indicate her parents and his parents, the 50% of the DNA that each of them received. And then in their child, the child will inherit 50% of his DNA from the father, 50% of his DNA from the mother, but they will be a mix of the paternal grandparents, the red and the purple, and there'll be a mix of the maternal grandparents, blue and yellow. Now, it doesn't always have to be this case. There are some instances where we see that, that the eggs and the sperm cells will retain a complete copy of one grandparent's chromosome, so they won't receive. So, for instance, maybe all of this would be purple, and they will receive their entire chromosome from just one of the paternal grandparents. But usually, this phenomena is observed only on the shortest chromosomes. But it can happen that it may have been that there was no recombination that occurred, and you only get one grandparent along that chromosome. Another really interesting fact that we've observed through testing people is that males and females have different rates of recombination. Males average about 27 crossovers per child, but females average about 41 crossovers per child. So what that means is we observe more crossovers between grandparents from DNA on the maternal chromosomes. And this is actually a nice example here on this picture where we see this, where there was just one crossing over period here for the from the father's side, where it changed from red to purple. And then over here on the maternal side, we actually see two crossovers from blue to yellow, and then from yellow to blue. And this is a pretty common phenomena where we see on the maternal chromosomes more points of crossing over than where we see on the father's side. All right, so the random nature of the places where crossing over occurs, now these places where the crossing over occurs, we call these recombination points, and we'll get back to recombination points. But it means that every child will be unique in how they inherit their DNA from their paternal and maternal grandparents. It's all random, and so some parts you're gonna one child will be red here, and then other parts maybe another child would get purple here on the top. And so because of the randomness of the inheritance of the DNA from the paternal and maternal grandparents, siblings only inherit about 50% of the same DNA. Now, I also want to add here that it's because of recombination, DNA inheritance is not so simple when we think about it from beyond the parent level. So we always talk about how a child inherits 50% of their DNA from the father and 50% of their DNA from the mother. That is true. But it is not the case that each child is guaranteed to inherit 25% of their DNA from each grandparent. Because of recombination and the random nature of it, it could be that you get a little bit more of one grandparent over the other, one like the paternal grandmother more than the paternal grandfather. And so we say that we inherit about 25% of our DNA from each grandparent, but it's not precise. It can actually be a pretty wide range, and we'll see it on the coming slides, how it can actually be larger or smaller than 25%, but it's about 25% on average. Okay, so that was the background on the biology of crossing over and recombinant chromosomes. Now, let's move from the textbook and trying to see it in real DNA testing and see how this applies in real life. Without further ado, let's look at DNA testing. So I always usually include this slide with my Y-DNA presentations where I give an overview of Y-DNA testing as well as autosomal DNA testing. Y-DNA testing, again, very briefly, is a very narrow test looking at inheritance of a male's Y chromosome from their direct paternal line ancestors. I do a lot of research into Y-DNA, but that will not be the focus of the discussion today. Autosomal DNA is the focus of, because in autosomal DNA, that is where we see recombinant chromosomes. So what is autosomal DNA? It looks at the ancestry inherited across those 22 pairs of autosomes. Because it's looking across all parts of your ancestors, both on your paternal side and maternal side, it does get diluted over time. So we just talked about inheriting about 25% of your DNA from each grandparent and about 12.5% DNA from each great-grandparent. As you get further up, second great-grandparent about 6.25%, and so on and so on. As you go further back in time, you inherit less of your DNA from specific ancestors, and you're less likely to find cousin matches who share that same DNA. And so usually with an autosomal DNA test, I like to say it's very useful for finding third cousins or closer. It can be used for finding more distant cousins, but it takes a little bit more work. And again, we're going to focus on this autosomal DNA when we talk about visual phasing. So with DNA testing and with autosomal DNA testing, you can take that at a few different places. Ancestry, MyHeritage, Family Tree DNA, 23andMe, all these places you can take DNA tests. And when you take a DNA test and you get your results, you will get a match list, and the match list will list, your all of your matches, usually starting from the ones where you share the most DNA. And the higher the amount of shared DNA that you have, which we call centimorgans, that's the unit of measure for shared DNA, or cm, that higher that total shared value, the closer the relationship is between DNA matches. So there's a great website called DNA Painter, and on DNA Painter they have this free tool called the Shared CM Project Tool. It's a great way to infer relationships based on the total amount of shared DNA that you share with a match. So I have here a nice little example. This is my paternal grandmother, Elizabeth Lehmann. She is listed as my grandmother, and she is my grandmother. And how much DNA does she share with me? Well, it's quite a lot. She shares 2,103 centimorgans with me. What you can do is you can type in that number 2,103 into here, and it will give you probabilities for what that relationship could be. In this case, it's very clearly that she is my grandmother. But they also have some basic information about what we tend to observe for certain relationship levels. So for a grandparent, usually the average shared DNA is 1,754 centimorgans, but there's a range. We've seen it as low as 984, as high as 2,462. So my 2,103 centimorgans falls in the middle of that. It falls in that range, but it's above the average. In fact, I inherited about 29% of my DNA from my paternal grandmother, which means I inherited less than 25% from my paternal grandfather. So this is a good example to show that you don't inherit, and actually here it says 30%. But yeah, high 20s, 30% of my DNA from my paternal grandmother. And it's a good example that you inherit about 25% of your DNA from each grandparent. It is not exactly 25%. So you can see the total amount of shared DNA, but we can really go further with the analysis and see where along your chromosomes do you share DNA with DNA matches. Now, unfortunately, most DNA testing websites, but not Ancestry DNA, and also now currently not 23andMe, but many DNA websites, especially MyHeritage or Family Tree DNA, they will allow you to further the autosomal DNA research by seeing exactly where the DNA overlap is between your matches and across all of the chromosomes. And the results can be given in a table format, but it can also be presented visually like we see here off to the left. And it shows, again, the precise segments of shared DNA, where they are found along the chromosomes, and the locations on all the chromosomes. So in this example, this is courtesy of MyHeritage. This is looking at, again, my paternal grandmother, Elizabeth Lehmann, and you can see the 29 DNA segments that I share DNA with Elizabeth all across the 22 autosomes. And you can see they're big stretches of DNA that I've inherited through the recombination, through crossing over and the recombinant chromosomes. These are all of the DNA I inherited from my paternal grandmother. It's quite a bit. We can see it again through this chromosome browser. Now, once you have a chromosome browser for DNA matches, and it doesn't just have to be a grandparent, it can be cousins as well, then you want to be able to create a chromosome map, which will trace the inheritance of your own DNA. So again, chromosome mapping, it's a technique used in autosomal DNA testing, which allows the tester to determine which segments of DNA came from which ancestor. To be able to build a chromosome map, I recommend you use the website DNA Painter. On their free tier, you can create one chromosome map, so you could do it for yourself. But if you're interested in doing visual phasing, which we'll talk about in a short bit, I do recommend that you pay for their higher tier, at least during the time that you're creating the chromosome maps. Once you you can create as many chromosome maps as you want once you are on the paid tier. And then even if you cycle off and you stop and you only pay, let's say, for 1 year, in year 2, you'll be able to keep all of the work that you've already done and build off of those. You just can't create any new chromosome maps. So I do recommend a DNA Painter, and the chromosome maps are one of the best features of the website. It has a lot of great tools for genetic genealogy, but chromosome maps are one of the best. So because one inherits a paternal and maternal copy of each chromosome, the mapping is only useful when mapping to the grandparent level or further back. So this is pretty important, but you don't want to, it would be pretty redundant to just say, oh, I inherited, all of chromosome 1 on the top from my father and all of chromosome 1 on the bottom here from the mother. Again, this is looking at each chromosome and looking at the father and mother's side. It doesn't really add much value to say, oh, this is all inherited from the father. We have to go back one step further to the grandparent level to see the segments of DNA inherited from each grandparent and to see those inheritance patterns, because they will vary across different children because of the randomness of recombinant chromosomes. Now, if you're like me and you're very fortunate to have some living grandparents, it can be pretty easy to map out your chromosome map to the grandparent level. So I have 3 living grandparents, and through these results, and including DNA inference in the case of my deceased grandparent, I have been able to map out 96% of my DNA, attributing my DNA to each of my 4 grandparents. So we can see this here visually off to the right. So the 96% of my DNA painted. The black is my paternal grandfather. The purple is my paternal grandmother, who we had seen on the previous slides. And then over here, the blue is my maternal grandfather, and the green is my maternal grandmother. And so you can copy, and paste just the segment information. There's actually no DNA confidential information that's held on DNA Painter. All you're doing is seeing where your shared DNA is and color coding it onto this chromosome map. And so this is very, very helpful to be able to have this map. And what I would just point out here is wherever the colors change, so from here from black to purple or from green to blue, these are instances where there are recombination points. Crossing over has occurred, and we have recombination points where the DNA switches between grandparents along the chromosome. So they'll either switch between paternal grandparents up top here, or over here between the green and the blue, they're switching between maternal grandparents. So here we can see visually what we just discussed about crossing over and recombinant chromosomes. This is a great visualization of the process. And it can be extremely helpful to have this map because I can now compare with unknown DNA matches and see where they fall along my chromosome map, and I can tell very quickly how they are connected to me. In other words, if they're only connected to me through my through a paternal grandmother, I can say, oh, I can see that, based off of the chromosome browser and where you map on to me, you must be a cousin to me through my paternal grandmother. And then that can help with finding connections with unknown DNA matches. So this is a nice final state. It's great. And again, pretty easy if you have living grandparents. But what happens if you don't have living grandparents and you can't just test them to see where you share DNA with them? Is there anything that we can do in this case? And that's where we get to visual phasing. And visual phasing is an alternative method for creating chromosome maps. When there are no living grandparents to test, we are really fortunate that there is this other solution called visual phasing. And so what is visual phasing? Visual phasing is an advanced genetic genealogy technique, and it's a process by which the DNA of siblings are assigned to their 4 grandparents using identified recombination points. And you do not need the testing of the parents or the grandparents. So it's a very powerful tool. But you do need to have at least 3 siblings to be able to do visual phasing. So that's the biggest caveat, is that you need to have 3 siblings. And if you want to do a full analysis, they have to be 3 full siblings. You can do a partial visual phasing analysis with half siblings, but again, you still need to have 3. Now, this visual phasing process initially does not automatically reveal which segment belongs to which of the 4 grandparents, but you then will match with cousins to provide this identification. I mentioned, again, visual phasing is best done with 3 or more full siblings. And it also requires having DNA from both known paternal and known maternal cousins. Visual phasing has been termed the ultimate genealogy puzzle, given how difficult and time-consuming the process can be. So I'm not going to sugarcoat it, especially the first time that you do this analysis. It can be cumbersome. Fortunately, there are some very helpful tools that make it easier. But once you get the hang of it, it doesn't get as difficult. Once you get the hang of it, it isn't as difficult as you think. And again, it's just immensely rewarding. It's almost like magic, being able to transform these siblings' DNA and build chromosome maps for them, even though they do not have any living grandparents for which we can readily test. So it really is magical, the result, once you're able to complete it or make a lot of headway with visual phasing. So now let's talk about it more specifically. We're going to go and see an example of how visual phasing worked. So back in 2022, I decided to embark on my first visual phasing analysis. This was for my father, Richard, and for 2 of his brothers, Leonard or Len and David. So I have 3 full siblings. They all took DNA tests: Richard, Len, and David. And again, they had taken DNA tests, so it made it easier. I didn't have to wait at all to be able to start this analysis. Now, if you look here, all 4 of their grandparents had long been deceased. Between 1950 and 1990, that's when all of their grandparents had died. So there was no way to have done autosomal DNA testing on the grandparents to be able to compare with. The only hope to be able to build this chromosome map more fully was to see how the 3 brothers inherited their DNA from the 4 grandparents and compare them to each other. So now let's take a closer look to see how this visual phasing process works. So I've tried to outline the steps broadly of how to do visual phasing, and there are some more links that I will include that people will be able to access so they can learn more. but I will go over these steps in more detail. So step number one, you need to have 3 siblings or more take autosomal DNA tests. So they have to have taken DNA tests for you to be able to do this process. And once they have taken these DNA tests, their DNA results need to be uploaded to the third-party website called GEDmatch. That's going to be a prerequisite for being able to do visual phasing, um, as we currently are able to do it. So step number two. Step number two is to use Stephen Fox's visual phasing Excel spreadsheet. This is a very handy spreadsheet that is accessible from a visual phasing Facebook group. So I have a link here that I will include to the Facebook group so you can join, and then you can download the latest version of the visual phasing Excel spreadsheet. It makes things so much easier to be able to run this analysis, but the only caveat for this Excel spreadsheet is that it must be run on a Windows setting, not on Mac. So you will need to have a Windows computer that runs Microsoft Excel in Windows in order to be able to run the spreadsheet. So once you get that Excel spreadsheet downloaded, you'll have to set it up, and you'll include the information from GEDmatch about the 3 siblings, and you'll pull all of this data from GEDmatch onto the spreadsheet. And it's very straightforward how to do that. And again, there is a walkthrough that the Facebook group has a PDF file that walks you through very thoroughly each step that's involved. But it is pretty straightforward to how to set it up. But sometimes it takes some time to pull all of this data and what it does, will compare the DNA of all the 3 siblings to each other across each of the chromosomes to see where they share DNA and where they do not share DNA. So you're comparing each sibling to another sibling and seeing where they share DNA and where they do not share DNA with each other. From this, and it's going to be presented visually, you can determine the recombination points, or we could also call them crossover points, for each sibling along the chromosome. So you'll be able to figure out where those recombination points are for each of the siblings. Once you solve for that important step of the recombination points, there is a logic puzzle that you need to solve to determine how each sibling inherited DNA at the grandparent level. Now, you won't know which specific grandparent yet, but you will know that there'll be one grandparent and the other grandparent, and one paternal grandparent and the other paternal grandparent, or one maternal grandparent or the other maternal grandparent. By the time, just by comparing the siblings, you'll be able to get to that step in most cases. And then step 5 is you'll utilize DNA matches of known paternal and maternal cousins to help assign the segments of DNA inherited to specific paternal and maternal grandparents. And then step 6 is you repeat all of these steps, 3 through 5, for all 22 autosomes. Each chromosome is its own separate puzzle to solve. So you'll get a lot, with just one visual phasing project, you'll get a lot of experience with visual phasing because you'll be solving logic puzzles for each of the 22 autosomes. So I wanted to dive in and give a specific example with the Lehmann brothers and how this works, how visual phasing works with this Excel spreadsheet. So this is going to be for chromosome 7. I thought this was a good example for illustrating visual phasing, and we'll walk through a lot of the key steps. So here we see comparisons between each of the 3 siblings and where they share DNA and where they do not share DNA. On this top area here, we see between Richard and David, denoted by R and D. On the second row, we see Richard compared to Len, R and L. And over here, the third one, we see a comparison between the brothers David and Len, denoted by D and L. And over here, we see the exact start and end places along chromosome 7 where they share DNA. We see the length of the shared DNA segment denoted in centimorgans. And obviously these are some pretty big segments here because they are brothers. And this is also the shared number of SNPs. These are the shared mutations in a row that correspond to this shared segment of DNA. So these boxes can be helpful later on when you want to put these into a chromosome map. But again, these help visualize the areas. Now, there are 3 different states for DNA inheritance. And I want to walk through each of them so you understand each of the 3 states that you're seeing. And that's going to be in these color coding regions. So the first state is if 2 brothers fully share a segment of DNA. That would be this blue line on bottom with a green line on top. And what that means, if they are fully shared DNA, it means the brothers inherited the DNA from the same paternal and the same maternal grandparents. That segment of DNA is from both, they both are from the same paternal grandparents and both from the same maternal grandparents. And so we see sections of this between Richard and Len over here, and we see it between David and Len over here, this blue and the green. Now the opposite is there are going to be segments of DNA where the 2 siblings don't share any DNA. And how is that possible? Well, it means that the brothers inherited DNA from totally different paternal and maternal grandparents. So, for instance, maybe Richard and David over here, they do not share any DNA. It's black with lots of, red lines on top here. That's a segment where they shared no DNA. Over in this area, it means that Richard maybe inherited his DNA from the paternal grandfather, and David would have to be from the paternal grandmother. And same thing, if it was maternal grandfather for Richard, it has to be maternal grandmother. They have no DNA in common. They actually got DNA from different grandparents, and so they don't share any DNA along this segment, along this portion of chromosome 7. And we see a lot of areas where there's black, where siblings do not share any DNA. And then there's a middle state where it's denoted by a blue line on bottom and then a mix of greens and yellows, usually more yellows, but there can be some greens, but not a sustained green. That is where 2 siblings partially share a segment of DNA. That means that only one of the grandparents they share, it can be either the paternal or the maternal, but they only share one grandparent. And for the other set of grandparents, they do not share DNA from. So we can see those areas. You can see big chunks here, here, and over here. All of this are just one grandparent shared between Richard and David. And we see this again on the between the other brothers as well. So it's important to know these 3 states because that will be very helpful with being able to solve the logic puzzle of visual phasing. So once you understand these 3 different states, what we are going to do is assign recombination points. So whenever the states of shared DNA switch, okay, and we can see that when the color changes across the chromosome. So whether it goes from black to blue here, or the blue with the yellow changes to blue to green, that indicates there is a recombination point for one of the siblings. So again, this is actually very important. Recombination points, they only happen to one of these siblings, and the task is to be able to identify across the entire chromosome where these recombination points are and who owns the recombination point. Okay? So in order to do that, you want to look closely at the same location across all 3 siblings to see who owns a recombination point. It's very clear that there's a recombination point right here, and I've highlighted it up close here. There's clearly a recombination point because it goes from black to blue, and here it goes from yellow to green. There's clearly a recombination point here, but the question is, who owns it? Who is the one who is changing inheritance from one grandparent to another? And how do we do that? You want to look. The owner of the recombination point is the one where is the common sibling where switching occurs. Okay, so let's look closely right here at this recombination point. Here again, the top row is between Richard and David, R and D. So on this top row, we see a switch between R and D. There is a switch where it goes from no shared DNA to partially shared DNA. And then if we go below, Richard and Len, R and L, they also have a state change from partial shared DNA to fully shared DNA. But if we look at the bottom here, there is no discernible change. They're really, for David and Len together, looking, comparing those two, it seems like nothing has changed. It's still partial shared DNA with some, yellow, mostly yellow and a little bit of green. So because the change occurred for where Richard connects with both, David and with Len, because it occurs with him, he is the common sibling where the switching occurs. It is Richard who owns the recombination point. And so what you'll do is you'll type in the letter R right up top here to denote that this is a point in time, point in the chromosome where the DNA is going to switch for Richard, not for Len, not for David. And at that point, it is going to switch for Richard between one of his grandparents. We don't know if it's a paternal grandparent, we don't know if it's a maternal grandparent, but for one of them, it's going to change. And so what you do is you're going to go through the first step, and this takes some time, is to go through each of the sections of DNA and assign recombination points across the chromosome. Now, some of them can get a little tricky, and chromosome 7 certainly had a little bit of that. As you can see here, there are actually 2 recombination points very close to each other right here. That was something I didn't figure out right away, but I was able to figure out with cousin testing. But here, instead of this being what looks like a recombination point for David, where it looks like it's going from blue to black and black to blue, both for the ones that share the common sibling as David, you would think that this is just one recombination point for David. In fact, it's actually 2 separate recombination points for Richard and Len. So there are some very tricky parts of it sometimes with assigning recombination points. It's important to be flexible and know that sometimes you may have to rework this as you go through the analysis. But in this chromosome 7, which is again one of the longer chromosomes, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 different recombination points across the siblings. Some are for R, some are for L, and some are for D. So they all have different places along the chromosome where they have their recombination points. And this is a critical first step for being able to solve for chromosome 7. So then, once you have these recombination points, it's time to solve to the grandparent level. And so what we see here is the end result of this stage. I do have a separate video that you can check out where you can see me actually solving for recombination points and for solving a whole chromosome to the grandparent level, where I walk you through this in more detail. But it is a logic puzzle where you start basically with areas where either siblings fully share DNA or they share no DNA. And so, and you label them with either G1 or G2 for one set of grandparents and G3 and G4 for the other set. We actually don't know when you're building this whether they are paternal grandparents up top or maternal grandparents up top, or vice versa. You actually won't know that right away, but you'll know it's one set of grandparents. It could be the paternal grandparents, could be the maternal grandparents, or G1 and G2, and one set of grandparents could be the paternal or the maternal. They are G3 and G4. And you can see just how this looks, right? This is a solved segment of DNA. This is between Richard and David, Richard and Len, and David and Len. And you can see that Richard and David don't share any DNA along this segment. And this translates to Richard having DNA from G2 and G4, but David has the opposite. He doesn't share any DNA. He inherited his DNA from grandparents G1 and G3, and they're all color-coded to show the difference. Now, but Richard and Len share fully all of their DNA along this segment, so lo and behold, this blue and green for G2 and G4 are exactly shared with Len in this area. So again, you use this logic puzzle to be able to fill in the gaps. The Excel spreadsheet actually helps you out a lot. Once you type in G1 and G2, it will fill in all the gaps up until a recombination point, which will help you fill out the map. And then you are going to combine all of those segments so that you can see this clear picture. And again, everything should check out logically that there are no inconsistencies. Everything looks logically correct here with this solution. So now, once you have it solved to the grandparent level and you don't know which grandparent, at this point you then want to introduce cousin testing to determine the precise grandparents. Now, it's important for cousin testing that these cousins, it's a little tricky because you need to find cousins who have taken DNA tests who only share one common grandparent. Now, if you have half first cousins, that's great because they will share just one of those grandparents. But otherwise, you're looking at someone who's at least a second cousin or more distant, maybe a first cousin once removed descending, but really a second cousin or more distant is what you're going to be looking for. And why is that tricky? Well, the further the relationship is, the less DNA you're going to share. And so second cousin is really ideal for this type of analysis. But as you get further away too, these are people who you probably are not as close with because they're a little bit more distant relatives. And so it might require some outreach if people haven't tested or if people have tested to be able to get to a chromosome browser either on GEDmatch or on other testing sites like MyHeritage to be able to compare the DNA to. And it really is a challenge to find cousins the more distantly you're related to be able to compare with these cousins. Now, you can compare with as many cousins as you want, but it's important to have at least one cousin from the paternal side and one cousin from the maternal side. You need that in order to be able to solve across the entire chromosome for the three siblings. So, after utilizing cousin testing, I was able to figure out that G1 was Landau, was their paternal grandmother. G2 became Lehmann. So G1 became this color blue for Landau, and G2 became Lehmann. So these were the paternal grandparents up top. I had assigned them correctly. This ended up being the paternal chromosomes, and you can see the inheritance pattern between the blue and the orange paternal grandparents. And then, for G3, for the maternal grandparent, that became Stein. That's this green over here. That became Stein. And G4 became Lichtenberg, which was the maternal grandfather. And this is the complete solution for chromosome 7, where they're assigned to specific grandparents for all three siblings across their entire chromosome. Let's walk through how this cousin testing works in practice so you can be able to figure this out. So I have a few examples here. This is the best example. This is, again, you just need to have one paternal cousin and one maternal cousin. This is a paternal cousin. Her name is Patricia Patton. Her maiden name is Lehmann, and she is a second cousin of the Lehmann brothers. And she's connected through their paternal grandfather, Leo Lehmann. So again, a second cousin. She's an ideal candidate for this type of testing. She had taken a test on Ancestry, and she graciously uploaded to GEDmatch, and I was able to pull that information from GEDmatch into the visual phasing worksheet. Now, it's most helpful if you're able to do that. You don't need to necessarily have this data into GEDmatch, but it makes it more visually effective to do so. So that's why all of the examples here are going to be for individuals who did upload to GEDmatch so that I could incorporate their data into the visual phasing spreadsheet. So this is looking over here in this bottom view for shared DNA between Patricia and the three brothers. Areas where there's blue are areas where she shares DNA. Of course, all this is just going to be partial DNA because she's not going to share DNA from both sides. This is just from one grandparent. And she descends from a brother of the grandfather, Leo Lehmann. Okay. We can see that these areas of shared DNA should correspond to the orange color, sharing that paternal ancestor. So if we take a look, we can see that for Richard, he has two segments of DNA that are shared. And again, some of these segments over here are quite significant. Many that are north of 20 centimorgans, one as big as 40 centimorgans in the case of Len. But each of these should map to the orange color, and lo and behold, they do, right? This segment and this segment that Richard inherited, they both correspond to Lehmann. Now, if we look at David, he didn't inherit any of these two segments that Richard did. And that's because this segment is a big segment he inherited from the other grandparent, from Landau. And that is why David doesn't share any DNA with Pat during this section. But there is a third segment over here that David shares that Richard didn't get. And we can see that this maps straight to the Lehmann ancestor, but Richard has crossed over, has a recombination point, and at that recombination point, he switched over to inheriting from the other grandparent. And that's why this third segment was not inherited by Richard. Now, who inherited the most? This would be Len. And I think this is probably a really powerful example. You can see 3 long segments of DNA that he inherited. He inherited the most with Pat and chromosome 7. But we can also see here very clearly the recombination points that occur for the other siblings. So for, in this case, especially for Richard. So Len actually inherited this whole segment of DNA from the beginning of the chromosome all the way to the 22.5 million part of chromosome 7 from his Lehmann grandfather. Now, Richard, though, only has a small bit of that 37 centimorgans. It's only 13 centimorgans that he's inherited. It's only the tail end. And before that, there's no DNA shared with Pat. So we know right at this point where it's switching from blue to black, this is a point where Richard has seen a recombination point on his paternal chromosome. It's switching from his paternal grandmother Landau to his paternal grandfather Lehmann. And we can see it starkly, so clearly by looking at how Pat compares to Richard and Len together, all together in this spreadsheet. We can see something very similar over here with the second segment where Len's got this very long segment that's 40 centimorgans. Richard's got one that's 26 centimorgans, but it ends right here. And if he had inherited from Lehmann for the entire way, he should look exactly like Len. But clearly there was a recombination point that Richard owns right here, and he's switching again between paternal grandparents, between Lehmann now to Landau. And he has no other recombination points along the paternal chromosome after this. So we can see how using these cousin relationships can help solve very quickly for the different grandparents. And what's important is that we're not just identifying segments that are from these specific segments. We can infer, because we have solved the logic puzzle, across the entire chromosome for all of the paternal grandparents. So even though we haven't even tested Landau yet, we haven't found a Landau cousin, we can infer that Richard inherited from his Landau grandmother in this section of DNA, because he didn't inherit the full segment that Len did. And we can apply it across the entire length of the chromosome. That's the power of visual phasing, that you're not just looking at smaller segments of DNA, you're being able to broaden it out to the entire chromosome and being able to infer inheritance at the grandparent level. Let's see another example. Now we're switching to the other paternal grandparent. Again, you don't only need to have one from each side, but the more the merrier in order to have confidence that you've executed this correct. So here we have Ron Rubin. He's more distant. He's a third cousin once removed of the Lehmann brothers, and he's connected through the paternal grandmother, Landau. So he is, part of this paternal grandmother, he's a blue cousin. So by comparing Ron's shared DNA with the brothers, we can identify all the segments of DNA on chromosome 7 that the Lehmann brothers inherited from Marie Landau. So we can see this result here, right? Ron shares no DNA with Len across this entire chromosome. There are 2 areas, though, that he shares DNA with Richard and David. Those must map to Landau. And again, some of these DNA segments are significant, especially this first one, 23 and 31 centimorgans. These are certainly real segments, but even the shorter one also aligns with the designation that we have. But we talked about how the beginning of the chromosome that Richard had to have inherited his DNA from Landau because Len had inherited from Lehmann. Well, here we see perfect example that he did, because Ron Rubin is a Landau cousin, and he inherited, and he's from the Landau side, and Richard happens to share DNA with him there, and so does David. Now we also see a clear recombination point taking place for Richard. And we can see this switching off between Landau and Lehmann taking place here. And again, I talked about how these two recombination points, they were really hard to decipher, but we actually know that there has to be a recombination point for Richard based off of this cousin match, that it can't just be David. Richard is clearly switching between Landau and Lehmann right here in the chromosome. Now, I just want to finish with just one other example from the maternal side. You need to have at least one paternal cousin, one maternal cousin. This is a maternal cousin named Charles Shreve Jr. He is a third cousin of the Lehmann brothers, and he's connected through their maternal grandfather, J. Chester Allen Arthur Lichtenberg. So here we have the shared DNA between Charles and the brothers, and he only shares DNA on chromosome 7 with two of the brothers, David and Len. It's one segment. It's decent length, it's about 14 centimorgans, and they need to map to the purple, to the Lichtenberg ancestor. So here we go. We see that they do map to Lichtenberg, and we know, it checks out right, that Richard wouldn't inherit it because he actually inherited that segment of DNA from Stein, from a different grandparent, and so this maps out perfectly with how we would expect. And all we need is one DNA cousin from the maternal side to solve across the entire chromosome. We know that one of the grandparents is going to be Lichtenberg, the other one's going to be Stein, and we can apply all those changes across the whole chromosome. So this is all we need in order to solve the remainder of the chromosome. So here is a look at what the final full solution looks like for chromosome 7, looking for Richard and David and Len from the beginning of the chromosome to the left to the end of the chromosome on the right, where they inherited their DNA with their paternal grandparents. That's the orange and the blue, Lehmann and Landau. And then for the maternal grandparents, the purple and the green, Lichtenberg and Stein. And you can see that, the inheritance patterns are random. It's very different. You can see that there tend to be more recombination points on the maternal chromosome, which is something we've talked about. Like for Len, he inherited this entire paternal chromosome from Lehmann. He had no recombination on chromosome 7 for the paternal side. But there are 2 recombination points for Len on the mother's side. And likewise, there tend to be, a bit more on the maternal side than we see on the paternal side. But for the other 2 brothers, they have recombinant chromosomes for both the paternal and maternal chromosomes, where they inherited some of their DNA of chromosome 7 from one grandparent and the other part of the DNA from the other grandparent for both the paternal and the maternal grandparents. So this is a nice way to just visualize the solution for chromosome 7. Another way to look at the visualized results is to also include these cousin matches and how they compare. So visual phasing will only get you to the grandparent level, but you can look at the specific DNA segments and infer more distant connections. So this is looking across chromosome 7 for Richard, David, and Len, across their paternal and maternal. This is in DNA Painter, and these are specific ancestors that they inherited DNA from. So for instance, for Richard, along this Landau side, he inherited a very big portion of this is actually from his great-grandmother, which was Marie's mother, Helena Rubin. And an even smaller segment of this is from Helena's father, Moses Rubin, and we can infer that from the DNA. And likewise, like on the mother's side, we have some DNA that is from over here we have Samuel. So this is the green is the grandmother, and this dark green is Samuel Stein. So this is actually the grandmother's father's DNA. We know that Richard inherited this part of the DNA from the Stein side, from the Bertha's father, Samuel Stein. And then we have another segment here that can be attributed to a different ancestor that's on the maternal, on Bertha Stein's mother's side, Ephraim Falk. And there clearly is a recombination point somewhere around here in between these two that denote this side that occurred with some of Richard's ancestors. But again, this is a way to visualize across the chromosome where they inherited DNA from specific ancestors. And Uh, in this map, we can see DNA inherited up to the second great grandparents. But for the full chromosome, you can see it at the grandparent level, and then for segments and for portions of the chromosomes, you can go even further back to a great grandparent level, to a second great grandparent, and beyond. So this is, as of October 2024, where we are in terms of Richard's chromosome map. And this is, again, what you'll do is once you solve for a specific chromosome, you'll take all those DNA segments data from GEDmatch and you'll copy and paste those and color code it into your DNA painter chromosome map. So this takes some work, and you have to solve for as many of the autosomes as you can. So we just talked about chromosome 7, but now this is looking at all of Richard's autosomes and looking across and seeing all of the inherited DNA. And again, visual phasing will help you to the grandparent level, and then you can find other cousins to fill in the gaps of the smaller segments to go further back in time. And you can see I have found segments of DNA that go up to the fourth great grandmother level for some of Richard's ancestors. And about 87% of his DNA has been painted. If we did not do this visual phasing process, the amount of DNA that would be painted would be much, much less, maybe as high as 30 or 40%, but usually not much higher than that. So the coverage of DNA inheritance goes up way higher by using the visual phasing process. And you can get a more complete picture of how someone inherited their DNA. And we get a pretty good complete picture. Now, I haven't completed everything yet. That's still a work in progress, like parts of chromosome 1 and some other chromosomes where I have not solved for either a paternal or a maternal grandparent, but I'm making progress and have solved for most of the chromosomes. This is what this looks like for David's chromosome map. You can see the colors look very different because he inherited his DNA differently, and the same thing for Lennon. All of them, I've solved for each of these brothers about 86 to 87% of their DNA currently as of October 2024. So I've just talked about visual phasing, how it works, and the lengthy process that it takes to be able to solve for each of the chromosomes. So why would I do all of this? Well, it's really cool to see the patterns of DNA inheritance, but it's even cooler to be able to use these built-out chromosome maps that is a result of visual phasing to be able to find connections to unknown DNA matches. So finding cousins, that's the biggest application of visual phasing. And it can be particularly helpful for those who are from endogamous populations, where you get a lot of DNA segments and you don't know if it's just endogamy or if it's like a real connection. Well, if you have a visual phasing mapped out, it becomes much easier to discern whether a match is real or is just endogamous. But it's useful. This analysis is useful across the board. It's not just for endogamous populations. But I have a good example here. So there's a DNA match on GEDmatch, who his name is Dave Rubin. I don't know anything really much about him, but just that he seemed to be a significant DNA match. What I did is I mapped his segments of DNA with each of the brothers, and look what I saw. It appeared that every single segment of Dave Rubin's DNA seemed to correspond with Blue, with Marie Landau, with the brother's paternal grandmother. It looked like that all of these shared DNA the brothers had inherited their DNA from their paternal grandmother, Marie Landau. And when you see all of these segments of DNA inherited, and this is the case so the gray here is Dave Rubin, the blue are all the segments from Marie Landau. When you see all of this overlap with just one grandparent, that is a very strong sign that the connection has to be through Marie Landau. And so I thought this was a very high quality match. And so what I ended up doing was building out a family tree for Dave Rubin. And it can be a little bit difficult because I didn't have much information to go on at first. But once I was able to figure out some of his deceased ancestors, I was able to build back his tree. And lo and behold, Dave was a relative to the Lehmann brothers, through the Marie Landau ancestor. And in fact, he is connected in 3 different ways, all through the Landau ancestor, but on both Marie Landau's father, the Landau side, and Marie Landau's mother, the Rubin side. There are three different connections. So this is a visualization of David Rubin building out his tree. And up here along his paternal Rubin line, we have three different ancestors, Asher Rubin, Razel Landau, and Maria Sadger. All three of these individuals are cousins from the Marie Landau. Asher Rubin is a connection through Marie Landau's mother, but these two, Razel Landau and Maria Sadger, they are through the Landau side. So Dave Rubin happens to be the Lehmann brothers' fourth cousin one times removed in two ways, and also their fifth cousin one times removed. Now, what's interesting is because there are all of these connections, David appears as a closer connection than he really is, because there's so many different ways in which he is cousins with the Lehmann brothers. This is why we see. The such a close DNA connection. So again, you can use DNA with unknown matches, and having the chromosome map built out through visual phasing creates a roadmap where you can get a real strong hint as to where a connection could lie, and it can help focus your research efforts to finding connections. Okay, the second application of visual phasing is about health characteristics. So this is one that had a lot more promise, I would say maybe 10 years ago, where the thought was there would be just point mutations. There would just be mutations that people have that would lead to health conditions. But we found that it's a lot more complicated than that, and there are usually a lot of different factors that both genetic and environmental that affect health conditions. But there are specific cases where there are mutations that lead to specific health characteristics. So I list two of them here that are known. And again, these are generally applicable. There are a subset of cases where it doesn't. But on chromosome 9, there's a section of the DNA where you inherit your blood type from specific ancestors. It's around 133 million from that marker on chromosome 9. And so for each of the brothers, I've listed which of their grandparents they inherited their blood type from. So for Richard, it was from both of his grandmothers. That was the subset of inheritance for his blood type. And then for David, though, it was for the grandmother on the mother's side, but the grandfather on the father's side. And you can see that. And that is on chromosome 9. Another area where you can see health characteristics, and this one I have found personally to be quite interesting, is eye color. That's right around the 28 million mark on chromosome 15. There's the gene OCA2, and there are point mutations there. It's a recessive gene that in 99% of the case, if you have a G from each parent, you will have some form of blue eyes. And so all the brothers have blue eyes, have a form of blue eyes. They both have a G from each grandparent, but we want to know which grandparent they received their G from. And so for Richard, we can see that on his father's side, it was his grandmother, Marie Landau. On his mother's side, it was his maternal grandfather, J. Chester Allen Arthur Lichtenberg. And you can see that this inheritance does differ between the three brothers for their eye color. Why do I find this so interesting? Well, if you're able to also pair this with cousin matches that go back further, you can trace where you got your eye color from to specific ancestors. For me, in my case, not from my Lehmann side, but from my maternal side, I've been able to trace my one of my G's for my eyes to a maternal third great grandmother who was born in the 1850s. Pretty cool to be able to make that connection. So again, you can apply some of this to infer health characteristics. The use cases here are pretty limited at this moment. Maybe as we go through more time, we'll be able to utilize the health data more fully. But it seems like a lot of the findings over the last decade is that things are a lot more complicated in terms of health characteristics. And you're not going to be able to necessarily use this data to learn a lot about health characteristics. You can learn some things through visual phasing. So in conclusion, the visual phasing analysis for these three Lehmann brothers has been a really big project, but it has been very successful, as I've now assigned north of 85% of the brother's DNA to their grandparents or even earlier generations. This research has led to the discovery of new branches of the family tree. It's made it much easier to determine how DNA matches connect with the Lehmann brothers. It also has the potential to yield insights into the inheritance of health traits and medical conditions. So, this visual phasing work will continue in the future, and I'll continue building off of it and refining them as more cousins are identified and the results are compared with the three brothers. And so with that, I will just conclude with a thank you. I want to give a big thank you to my father and his two brothers, David and Len, for taking the DNA tests and for uploading their data to GEDmatch. It was only thanks to their DNA tests that I was able to do this analysis of visual phasing. I also want to thank the many cousins who have taken DNA tests, and especially those who have uploaded their results to GEDmatch. This has really helped drive the visual phasing project forward. It's taken a lot of work, but we've made a lot of progress at the, a couple years later. So I'll just conclude here with an appendix. There are a couple slides here that I'll mention. Just this is a really nice slide that has some of the genetics terms. We talked about the biology, but just being able to have definitions for some of the terms that were discussed. And then this is one that I'll conclude with, and this is just looking at, through DNA Painter, the specific cousins that contributed to figuring out the DNA inheritance across chromosome 7. And you can see there are paternal cousins and there are maternal cousins. And I want to thank all of these individuals for their help with furthering this research. And with that, I will conclude. I will say, for those again who want to have a deeper look at how I solve one of these chromosomes, I highly encourage you to check out my other video where I solve a chromosome, figuring out recombination points and assigning them, and figuring out to the grandparent level, and then using the cousin matches, so you can see all of that. And I hope that many of you get inspiration to do your own visual phasing analyses, and because it can be immensely rewarding and can really take your genetic genealogy skills to the next level. Thank you so much.